This tutorial introduces you to enzymes and how they work. If you like what you see on our channel, please like, share, subscribe and turn on notifications. Within every living organism, chemical reactions take place all the time. These reactions happen too slowly to keep organisms alive unless they are speeded up by catalysts. So what is a catalyst? A catalyst is a substance that increases the speed of a reaction without being changed or used up in the process. In our body, almost every reaction is controlled by catalysts called enzymes. Enzymes are protein molecules found in cells that function as biological catalysts or biocatalysts. That is, it speeds up a chemical reaction and is not changed by the reaction. They work by lowering the activation energy, that is, the energy required to make substances react, which in turn ensures that the reactions are completed at a faster pace. Enzymes are named according to the reaction that they catalyze using the suffix ase. Enzyme amylase breaks down starch into maltose. Maltase breaks down maltose into glucose. Sucrase breaks down sucrose into glucose and fructose. Protease breaks down protein into amino acids. Lipase breaks down lipids into fatty acids and glycerol. Lactase breaks down lactose into glucose and galactose. Catalase breaks down hydrogen peroxide into water and oxygen. Now let us understand the properties of enzymes. All enzymes are proteins. Enzymes are specific, which means that each kind of enzyme molecule has a shape that makes it suitable for catalyzing only one kind of chemical reaction. Enzymes are catalysts. They are not changed in a reaction and can be used again and again. Enzymes work best at a particular temperature and pH. Enzymes can catalyze different types of reactions, breaking large molecules into smaller ones. For example, in the process of digestion, large food molecules like starch are broken down into smaller sugar maltose by the enzyme amylase so that they can be absorbed and then used. Building up large molecules from small ones, for example, the enzyme starch phosphorylase builds starch molecules from small glucose molecules inside plant cells. How do enzymes work? An enzyme works by allowing the molecule of the substance on which it is acting, called substrate, to fit into it. The reaction takes place on a part of the surface of the enzyme called the active site. Only a substrate molecule with a shape that fits into this active site will take part in the reaction catalyzed by this enzyme. Other substrates have the wrong shape to fit in and will not be involved in the reaction. This type of a mechanism is called the lock and key mechanism. Here, the enzyme molecule is like a lock. The substrate molecule has a complementary shape to the enzyme and can fit into it like a key. The substrate binds to the enzyme at the active site, forming an enzyme-substrate complex. The enzyme then slightly stresses or bends the substrate, splitting it into two product molecules that drift away from the enzyme molecule, leaving its active site free to operate again. Enzymes can also help build large molecules from smaller ones like this. Now there are many factors affecting the enzyme action. Stay tuned as we understand this concept in our next tutorial.